Alright, so now we will go on to 3.5, the plant hormone. So this is the last one, okay, on the chapter 3. Okay, last one in chapter 3. So chapter 3 we will see the plant hormones also, right? After we have seen the human hormone and how the homeostasis, the coordination of the osmotic pressure, okay, in the kidney. So now we will see the plant hormone. So how the plants regulates, okay, how the plant regulates the process that happen inside the body. So it's 3.6, huh? 3.6. So let us see. So in this chapter, yeah, if you have read up your textbook, okay, we will only focus on auxin and ethylene. Okay, there are two hormones that we will focus in, auxin and ethylene. But if you go online, okay, there are more. There are about 4 or 5, okay, plant hormones that is mentioned or is known. Okay, people have do research and they have found a few types of plant hormones that is available or is there in the plant. Okay, so we will only focus in auxin and ethylene. Okay, later I show you a video. The video will talk about other hormones also. Okay, now that auxin that we can see, yeah, okay, in your textbook, okay, auxin is actually the main hormone that is found in the plant. Okay, and these hormones are responsible for the cell elongation. Okay, so cell elongation, it will make the plant grow taller lah, in another word. Okay, grow taller. And then beside of that, okay, okay, it also will trigger the growth of adventitious roots. Adventitious root means the branches, okay, the root that is not the main one, not the tap root, okay, the roots that grow out from the tap. Okay, or for example, let's say you have a branch and you want to stimulate growth, the growth of the roots. Okay, we can also treat the plant with auxin, okay, to promote growth of the roots, okay, the root hairs, okay, root hairs. Another function of this hormone is to stimulate parthenocarpy. So parthenocarpy is a term we use when the fruit is produced without fertilization. Okay, parthenocarpy is a production of fruits, okay, of fruits without fertilization. Okay, in chapter 4 you will see that how the fruit is produced, or even in your lower form, you have seen that fruit is produced when after fertilization. After fertilization also only we will get your fruits. Okay, but this auxin has the ability to stimulate parthenocarpy, means you can get fruits without fertilization. Okay, another hormone that we have here is ethylene. Okay, ethylene is an alkane, and this is a gas type of hormone and very special. Okay, that means you won't see the hormone, okay, because the hormone is in terms of gas. Okay, and then the taste or the gas will have some smell, the sweet or the mosquito, or this one is actually too easy. It's the smell of the fruits. Okay, sometimes when you see the fruits is ripe, okay, when the fruit is ripe, so you will have that kind of sweet smell. Okay, that smell, the odor that smell good or smell sweet, okay, that is ethylene, the gas, the hormone. Okay, so in agriculture we will use it, or we can produce alkene artificially, means chemically, okay, in the factories to force the ripening of the fruit. Meaning if the fruit is not ripe, or it's not it take longer time to ripe, okay, then we can use ethylene and then we make or force the fruits to ripe. Okay, another function is to stimulate the opening of the flowers, and also it will cause abscission. Abscission means the leaf turns yellows and then drop. Okay, the shading of the leaves. Okay, this ethylene will cause this thing. So normally ethylene is produced a lot, okay, during after, I mean, some autumn, okay, during autumn. Okay, so we will see one by one. So how the auxin, okay, function. Okay, let us see here. So the site of production, okay, where it is produced. Okay, so auxin is produced at the tip, okay, at the tip here and at the tip here. Okay, actually yeah, it's at the tip lah. Okay, so on the shoot or on the roots. Okay, and also one thing about auxin, it is a chemical, and this chemical, it can diffuse from high concentration to lower concentration. So it's produced here, but it needs to do with function, it will go down or will diffuse down to the zone of elongation. So if I have a shoot here, okay, we have zone of division, zone of elongation, and zone of differentiation. So there are three types of zone. Of course, it's on the tip of the shoots. And also for the roots, at the tip of the roots, there are three zone also. Okay, the first zone, division, second is elongation. So the hormone is produced at this zone, okay, zone of division, and then it will be diffused, okay, to the zone of into the zone of elongation. Okay, so what it do? So auxin can be measured in different concentration, yeah. So at the roots, to stimulate growth, you see, this is the concentration needed. Okay, the concentration between -6 to -4. Okay, concentration of auxin. If you have too much of auxin in the roots, it will inhibit the growth. Okay, so the concentration needed in the roots and needed at the shoots is different. Okay, so when they will produce accordingly lah. Okay, but when we do experiment, we can alter or we can change the concentration to show how it stop or inhibit the growth at the roots, and then to show how it stop or promote. Okay, it's not stop, it's how it promote the growth at the shoots. Okay, we will use the auxin produced at the tips, okay, to do the experiment. Okay, so this graph is just to show you the different concentration needed, okay, at the roots and at the shoot for growth, for become longer. Okay, for become longer. So roots will need lower concentration, shoots will need higher concentration. Okay, so this is another graph, also the same. So the B is the stems, the B is the roots, okay, the A is the stem. Okay, so that means there are different concentration. So if I use same concentration, for example, I use a concentration of 10 power of 6 at the roots, yes, it will start become long, okay, grow. But if the 10 power -6 concentration is used at the stem, doesn't have any effect. Okay, that is what we can explain. Okay, that is how we explain. But let's say I use 10 concentration or 1. Okay, the concentration of arsen is 1 ppm, yeah, 1 ppm. So if I use 1 ppm at the roots, okay, the root will not grow. Okay, so that is how we explain. Okay, like we see experiment. So experiment that we want, that we need when we want to do experiment on auxin, yeah, we have to get the auxin first. Of course we can get it outside chemically, but let's say we want to get it from the plant itself, so we can do like this. Okay, so this is the plant. Okay, this is the plant. So I just cut, okay, tip of the collector. Collector means the part where it produce auxin. Okay, the tip. Okay, so I cut here, and then I put it with okay, I put it on the jelly. Okay, I put it on jelly. So the first experiment is I cut away this and I don't put anything. Okay, the second experiment is I cut away the collector and then I put on the jelly, and then instead of putting back the collector, I put up the jelly. Okay, and the third is I put the on the jelly, on the agar, yeah, on the agar, and then the agar I put one side only. Okay, you see half of the shoots only. Okay, so we see what is the difference. This one is to show that auxin is produced at the tip, yeah. So first experiment, I remove the tip. Okay, so after I remove, after some days, there is nothing. Okay, no further grow. Okay, no further grow. Okay, but this one, after that I put the agar back on top. After 2 hours, I put the agar on top, and then we can see that after a few weeks the plant grow up. Okay, there's no result showing here. Okay, no result showing, but we can say that the agar, when it's placed on the cut collectel, okay, this collectel will grow up. Okay, or the shoot will grow up. Okay, then as for the third experiment, okay, if I put one side, okay, after few days, okay, the coletel will grow towards the parts towards the other part of the agar, okay, or the part where we put agar on the left, right, so it will grow to the right. Okay, if put on the left, it grow to the right. Okay, why or how to explain? Okay, so this one no problem, because the second experiment shows that auxin is produced at the tip and it is grow upward. Okay, it grow upward when the agar is put on or placed on top. But the third experiment, okay, the third experiment, how come it grow towards the opposite direction of the block? It shouldn't. It's something that is something that can be explained, okay. We have to explain that one. Okay, let us see. So we see the experiment how to get the auxin from the coletel. Okay, so this is the tip. Okay, we can remove and then put on the agar 4 hours. So after 4 hours, actually auxin diffuse into the agar. So diffusion happen from high concentration to lower concentration. Okay, so from high concentration to low concentration at the block. Okay, so if I put the block on top, I can actually, the agar now become the one with auxin. Okay.